Hello and welcome. Today let's discuss Chapter 17, which takes us into the nervous system. This is a very fascinating system. We'll see the nervous system along with the endocrine system coordinates and regulates the function of other body systems. Now, our nervous system in most animals, including humans, is going to be divided into two anatomical divisions. You have number one, our central nervous system, or it's abbreviated as the CNS. And number two, we have our peripheral nervous system, which is abbreviated as the PNS. Now here, when we go through and we talk about the nervous system, as you can see, we'll start off with nervous tissue first, and then we'll progress through and get the whole system taken care of. Now again, the nervous system is divided into two anatomical divisions. We have the central nervous system, we said, or the CNS. And the central nervous system, or the CNS, is going to be made up of the brain and the spinal cord, as we can see. Then the peripheral nervous system we mentioned, the PNS, is made up of everything outside the CNS. Everything outside the CNS. So we'll go through and we'll check that out. It'll include ganglia and it'll include, we'll see, nerves as well. Now when we talk nervous system, it's made up of two types of cells. First we have our neurons. I like to consider them the VIP cells. However, they can't survive without neuroglia. So equally as important as well. When we talk neurons, neurons are going to transmit our nerve impulses. They transmit action potentials, we refer to nerve impulses also as. And our neuroglia, they're going to be responsible for providing our neurons with support and also nourishing them. Now, when we go through and we talk about our neurons, we have three classes of neurons. We have sensory neurons, we have interneurons, and we have motor neurons. Our sensory neurons, they're responsible for routing messages towards the CNS. They take our messages towards the CNS. And they may have specialized endings that we call sensory receptors. Our interneurons, we can see second, they lie entirely within the CNS. And they're going to receive input from the sensory neurons and other neurons. And then what they do is they communicate with our motor neurons. Our third type of neuron are our motor neurons, and they carry messages from the CNS now to an effector. To an effector, when we talk effectors, those are going to be our muscles and glands. So here let's look at all of that. So first here we can see we've got our spinal cord, and then we can see it's going to be communicating with the brain, and the brain will be communicating with it, and they make up our CNS, our central nervous system. And everything outside of it, we said, is our peripheral nervous system. First here we have our sensory neurons. Now our sensory neurons, we said, are going to be responsible for sending information to the CNS. So we can see that arrow is pointing that out to us. And here you can see these neurons, they have their cell body outside of that CNS. So that cell body is in the PNS. Then we talk our motor neurons. Our motor neurons, we said, are going to be sending information away from the CNS to our effector organs. So here we can see one example, our skeletal muscle. And now here when we look at our motor neurons, you can see their cell body is in the CNS. Their cell body is in the CNS. And third, here we can appreciate our other motor neurons, which are going to be motor neurons for smooth muscle, cardiac muscle, and our glands. Here you can see they have a junction in between two neurons. The first neuron, its cell body is in the CNS, and it will form a junction with another neuron, which is going to be responsible then for taking the message to each of those different effector organs. Now, when we talk about our neurons are going to have three main parts to them. We can see they have first a cell body. The cell body is also known as the soma, S-O-M-A. And it's going to contain the nucleus and other organelles. We can see that here. When we look at this picture, you can appreciate the nucleus inside the cell body. Same thing inside of this cell here. And the same thing here with this cell. And the same thing here with this cell. So that's kind of something you can notice right off the top. So here then let's look at the three parts of a neuron. And we can actually draw that out here as well. Then we talk about the three parts of a neuron. First part we're going to have, we can see, is going to be the cell body. And when we talk about the cell body, you can see here we've got a cell body. And then here we can see the cell body is going to contain the nucleus. Along with that, it'll have the other usual organelles that are going to be found inside of the cell. Okay, so here you can see and other organelles, my little cheap example of other organelles. And then here, the second part, we can see our dendrites. Now, when we talk dendrites, dendrites are going to be extensions that receive signals from other neurons. So here we can appreciate that very nicely. And then, so here when we see extensions that receive signals from other neurons, so if we have another neuron, we're going to be able to draw that in here in a few minutes, and we'll see how that happens. Next, then we have our axon. When we talk axon, axons are going to conduct impulses away from the cell body to other neurons or effectors. So here we can see an example of a neuron. Here we've got our dendrites. So here we can draw that out, dendrites. And then here we've got our cell body, the structure right in here. And the last is our axon we see right here. So cell body contains a nucleus and other organelles, we said. Now dendrites are extensions that receive signals from other neurons. So if we were to draw out another neuron, so here we said we'll draw out another neuron. So here we can see we have the cell body, a whole bunch of dendrites. Okay. And then here is its axon. Its axon. So we'll draw its axon. And here we can see then when this cell stimulates the next cell, okay, the signal is going to come in here on the dendrite and it's going to travel towards the cell body. Travels towards the cell body, the signal does, and the incoming signal then gets integrated by this neuron. And if that signal is strong enough, this neuron then will send out a signal of its own, and that signal is going to be sent, you can see, away from the cell body along that axon towards the end of the axon. So here we can see dendrites are extensions that receive signals from other neurons. So we saw that here with this dendrite. And then our axons are going to conduct nerve impulses away from the cell body towards other neurons or effectors. And we can see that here with our axon. So next here we can see better drawings of neurons. Now here we can see we've got first a sensory neuron, and then we've got our interneuron, and then we've got our motor neuron. Now here let's identify all the parts we just learned about here first on this motor neuron. You can appreciate the cell body, nucleus inside, the nucleolus, and then all the usual organelles you can appreciate here on the sides. And then here are all of its dendrites. And here we have the axon. Second, here we can appreciate the interneuron. The interneuron, now same thing, soma, cell body, the usual organelles, and the nucleus. And then here we've got our axon. And here we've got many dendrites. And then last here we can see our sensory neuron, which is going to be responsible for sending impulses then towards the CNS. So here we can see these impulses arrive here onto the interneuron and onto its dendrite. And then the interneuron will process that information, and then it'll send out a response via its axon. And here we can see the next neuron, its dendrite, is going to be communicating with the axon of the interneuron. So this dendrite receives the signal from the interneuron. The cell body will process that information, and then here we can see its axon is going to help spread the nerve impulse. Now here we can see on this axon we've got myelin. We've got myelin. Now in the peripheral nervous system, we'll see myelin is going to be produced by Schwann cells. Myelin is a lipid-rich substance, and it's going to wrap around axons to act as an insulator. Now, in between two myelinated regions, we have a node of Ranvier. Node of Ranvier is going to be a gap in the myelin sheath, basically. Now, when we talk myelin in the PNS, we'll see it's going to be important in nerve regeneration. Next, then we can see in the central nervous system. Now, in the central nervous system, myelin is going to be formed by oligodendrocytes. And we have little nerve regeneration in the CNS. So here we can appreciate an axon, and here we've got myelin produced by this Schwann cell. And here's the Schwann cell's nucleus. So the Schwann cell wraps itself around one segment, produces the myelin, And then here you can see the next Schwann cell does the same thing, as does the third one. And then in between these cells, you can appreciate the nodes of Ranvier. And they're important because what they're going to do is the action potential, the nerve impulse that's traveling along this axon is going to get regenerated here. Here in the myelinated region, the action potential is going to be able to spread without having to be regenerated. So here we don't have any myelin, here it has to be regenerated. So then it takes off in the next segment, and then the next node of Ranvier, we have to regenerate it again, and then it takes off in the next segment, and that continues all the way through. So when we talk central nervous system, I would like you to know the central nervous system is going to be composed of two types of nervous tissue. We have gray matter and we have white matter. Gray matter is going to contain cell bodies and short fibers, while white matter is going to contain myelinated axons that are going to run in tracks. So when we talk gray matter, you can see gray matter is going to be found making up the surface layer of the brain and also the central part of the spinal cord. Now when we talk white matter is going to be found located deep in the brain, and it surrounds gray matter in the spinal cord. All right, next then let's move on to transmission of nerve impulses. Now the nervous system, we said, is going to use nerve impulses, or we call them action potentials, to convey information. Okay, it's to convey information. Now studies on the nature of a nerve impulse have been done using excised axons and a voltmeter. Next here then we have the term voltage. Voltage is going to be expressed in millivolts, and it is the measure of the electrical difference between 2 points. In neurons, the 2 points are the inside and the outside of the neuron. Now, before we jump into the action potential and see how the nerve impulse is going to be transmitted, first let's talk about the axon in a resting state, and we'll look at its resting potential. Now, when the axon is resting, it has a potential basically. When an axon is not conducting an impulse, the voltmeter recorded a potential across the axonal membrane that was equal to or about minus 70 millivolts. When we look at the inside of an axon, we'll see the inside is negative compared to the outside. So that's why we have the minus 70 millivolts. Called the resting potential because the axon is not conducting an impulse. And it's going to be due to an unequal distribution of ions across the membrane. We have more sodium outside than the inside, and we have more potassium inside compared to the outside. Our resting potential, we'll see, is going to be maintained by our sodium-potassium pump. So if sodium happens to leak in and potassium leaks out, these active transporters are going to pump sodium out and pump potassium in. The membrane is somewhat permeable to sodium and potassium. They tend to diffuse towards lesser concentrations. And then that's when that sodium-potassium pump kicks in. The polarity of the membrane is going to be based on having more positive ions outside the membrane than compared to the inside, because the inside, we said, is minus, it's negative. And the inside is going to be largely negative because we have large negatively charged organic ions inside. So here we can see the axon's membrane in a resting state. In the resting state, we can appreciate the high quantity of potassium ions inside the axon versus the outside where we have a high quantity of sodium ions. And here we can see all of our gated sodium and potassium channels are closed. So here then in the second state, we can see we've got open sodium channels. And these open sodium channels are going to allow sodium to cross. And we said sodium is going to rush into the cell. So when sodium, which has a positive charge, begins to rush into the cell, what's that going to do to the voltage? It's going to make it less negative, taking it more towards the positive aspect. So you can see here in the graph, we're going from minus 60 millivolts up towards minus 55 millivolts. Minus 55 millivolts, we can see here, happens to be threshold. So if the stimulus causes the axon to reach its threshold, and if that threshold is reached, then an action potential we'll see is going to be generated, and that's going to cause the voltage to go all the way up to plus 35 millivolts. We have a change of about 100 millivolts take place. So here you can see, next then we've got the state of depolarization, and then here we've got repolarization. So here's where the action potential is full on going. The impulse is actually being generated. So let's talk more then about action potentials. So when we talk action potentials are going to be a rapid change in the polarity across the axonal membrane as an impulse occurs. So if we go back to this picture, here in this bottom picture where you have the graphic depiction, you can see they're showing you the cell in the resting state, it's at minus 70 millivolts. And if an impulse comes in, that impulse, if it's strong enough, it can take us to threshold, where then an action potential will be generated. But now if that threshold is not reached, so if that incoming impulse is not strong enough to take us to threshold, we'll see we'll go back to resting and the cell will maintain the resting state. But we're not going to maintain the resting state. We want to see that action potential. So here then when we talk action potential, we said it's a rapid change in the polarity across the axonal membrane as impulses occur. Now first we have what's called the all or none phenomenon. This is basically telling us that if we hit threshold, we're going to have an action potential. We're not going to hit threshold and say, oh yeah, we just had half of an action potential occur. No. It's the action potential is going to occur completely or it doesn't happen at all. So that's the all or none phenomenon. So the action potential is going to occur if a stimulus causes the axonal membrane to depolarize to a certain level. We said that's called threshold. The strong stimulus does not change strength of the action potential. The action potential is going to remain the same strength. It causes the voltage change to happen to about plus 30. It's a voltage change of about 100 millivolts, we said. So that quantity of voltage change is not going to change. So even if you have a strong stimulus, again, it does not change the strength of an action potential. Merely changes the frequency of firing. So instead of having an action potential that goes up 200 millivolts, no. We're just going to have more action potentials generated if we have a stronger stimulus. Now when we talk action potentials, they're going to require two types of gated channel proteins. Sodium gated channels we saw, and potassium gated channels. Now when the sodium gates open, sodium starts to flow down its concentration gradient into the axon we saw. Now here we can see when membrane potential changes from minus 65 millivolts to about plus 40 millivolts, this is going to be called depolarization. Now when the axon's membrane was in a resting state, we refer to that as being polarized. Now here, when the voltage is changing from minus 65 millivolts to about plus 40 millivolts, the cell is undergoing depolarization. So it's called depolarization because the inside changes from a negative to a positive. Then we'll see the potassium gates are going to open. When the potassium gates open, potassium is going to do the same thing. Potassium is going to flow down its concentration gradient and it's going to make its way out of the axon. This helps to bring the potential back down to minus 65 millivolts, right? Because we took the cell to plus 30 millivolts, right? To plus 30 millivolts. And now what we do is we remove potassium ions. Potassium has a positive charge. So if we start to remove the positive charges from the inside, that's going to take the cell back down to a negative charge. So it brings the potential back down to minus 65 millivolts. And now this phase is called the repolarization phase because it helps the cell return to its original negative polarity as potassium exits the axon. So here we can see in this corner picture, you've got the first phase, that's depolarization, and it tells us there sodium moves to inside the axon. So when sodium moves to the inside of the axon, sodium has a positive charge, it's going to cause the inside of that axon to become more positive. And then the sodium channels will close. Potassium channels open up, we said. And here then you can see this will allow us to enter the repolarization phase. With potassium channels open, potassium is going to rush out of the cell, and this takes the positive charge out of the inside of the cell, bringing us back down to our negative reading of minus 65 millivolts. So we're back at our resting potential where we said the cell is polarized. It just underwent depolarization and a repolarization to become repolarized again. So that's one action potential, that's one signal. So we can see here this voltmeter recorded this action potential, this voltage difference, as it made its way across that axon. So action potentials are going to be a rapid change in polarity across the axonal membrane as impulses occur. So let's continue forward. So we said sodium gates open first, and then potassium gates are going to open, leading to repolarization. So we can see this action potential is going to happen over and 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 over again, all the way from one end of the axon to the other end of the axon. So this is how nerves send signals. Now, when we talk about the conduction of an action potential, we have to look at a couple of different scenarios. First, in a non-myelinated axon, action potentials are going to travel down an axon one small segment at a time. You can think of it here like a domino effect. So one segment will happen where one domino will fall, and then the next segment, the next segment, the next segment, and action potential is going to make its way all the way across. As soon as an action potential has moved on, the previous section undergoes a refractory period. Now, during this refractory period, the sodium gates are not able to reopen. So if they're not able to reopen, we can't cause a cell to become depolarized, and an action potential just can't occur. The action potential also cannot move backwards. Instead, it always moves towards the axon terminals, the ends of the axons. When the refractory period is over, the sodium-potassium pumps are going to restore the previous ion distribution by pumping sodium outside the axon and potassium back inside. Now, when we talk in myelinated axons, you'll see the gated ion channels are concentrated in the nodes of NVA. So therefore, we're going to see exchange of ions is only going to occur at the nodes. And this is going to result in a faster action potential than in a non-myelinated axon. Because here what happens is you have basically a skipping or a jumping type of pattern. The action potential jumps from node to node, as we said, and that's going to be called saltatory conduction. Saltatory conduction. So let's jump back. So here we can jump back and we can see that right inside of here. So we said the action potential is going to get generated in here, and it's able to travel all the way across this area that's myelinated, but then it has to be regenerated here again. So it's kind of like a skipping, you can think of a jumping pattern. Saltatory conduction. Now if there's no myelin, right, the action potential has to get generated over and over and over and over and over again, all the way through until we reach the axon terminals. So you see here why it's faster. Now, if we have an axon that's fully myelinated, that's going to spread the signal the fastest because the action potential is going to occur at the beginning, and the myelin is going to help to then spread that signal all the way to the axon terminal. So you can see we have three different types of conduction down our axons. We have in a non-myelinated, and then we have myelinated, two types, fully myelinated, which is what we'll see with some axons. And then we have our saltatory conduction. So now let's move through and let's check out the transmission across the synapse. So we saw a message basically make its way into the dendrite, go into the cell body, from the cell body make its way down then the axon. Now from this axon it's going to cross the synapse, basically it's going to go from one cell to another is what we're going to see here. So we saw that before. We said where you're going to have the axon ending of one cell, one neuron, is able to communicate then with the dendrite of another. So that's where we're at now. We're at the synapse. Now we're going to see how one cell is going to communicate with the next. So here we can see now every axon branches into many fine endings. So let's look at that. Now what we drew in before is we had a neuron that looks something like this. We had the dendrites, right? And then we had a cell body with the nucleus. And then here we had an axon. Now if we were to look at this axon now in greater detail, this is what it would look like in greater detail. We would have the axon coming down like this. And then here we could see the axon would make its way like that. This is what those branches would look like. Now these branches, if we were to look at in greater detail now, you can see full on, they're going to have these swellings. They have these swellings here. It doesn't look, it's not supposed to look so like a heart. It's supposed to be a little bit more rounder. So let me try to do this one here a little bit better. So here we can see it'll look like this. Kind of like a flask like. Okay. So here then you can see this is going to be the axon terminal. So each is tipped by a small swelling, which is called the axon terminal. So here you have then the axon terminal in greater detail. Now when we talk synapse, a synapse is going to be the region where each terminal lies close to either the dendrite or the cell body of another neuron. So if we were to draw now another neuron into here, we would draw like the cell body, let's say here, we can have then the dendrite of one neuron, of this other neuron right here, and then here we can see the axon then of the next neuron. Now this area right in here is the synapse. So again, the synapse is the region where each terminal lies close to either the dendrite or the cell body of another neuron. Now here, coming back to this picture, right inside of here, we saw right in here you have one synapse. So right in here we have one synapse. And then right here we have another synapse. And then right in here we have, you can see 1, 2, 3, 4 different synapses right up in there. So again, it's a region where each terminal lies close to either the dendrite or the cell body of another neuron. Now, we have here the synaptic cleft. The synaptic cleft is a tiny gap. It's this gap you see right in here. It's this tiny gap that separates the two neurons at a synapse. Now, here we're going to add some terms here as well to the neurons. Now, this synapse we see here has two neurons. So here is our presynaptic neuron. This neuron now is the first neuron in this order here, basically. And then you've got the postsynaptic neuron. Here then we have our postsynaptic neuron right inside of here. We'll label it as number 2. So here we've got number 1, the presynaptic neuron, and then we've actually got the synapse, and then we've got the postsynaptic neuron. Next, then let's continue forward. Now when we talk transmission across the synapse, we'll see communication between the two neurons is going to be carried out by molecules that we call neurotransmitters. These neurotransmitters are going to be stored in synaptic vesicles, and they're able to cross the synaptic cleft. So right in here now, if we were to look in greater detail, we would have these little vesicles, and these vesicles are going to have many neurotransmitters in there. So right in here we can draw, let's just draw one big synaptic vesicle in here, and you can see all these will be neurotransmitters right inside of there. So again, communication between two neurons is carried out by molecules called neurotransmitters, and the neurotransmitters are going to be stored in synaptic vesicles. So here you see they're stored in these synaptic vesicles in the axon terminals of the neuron. These neurotransmitters are able to cross the synaptic cleft, and they're going to be released when action potentials, when the nerve impulses reach the axon terminal. You see why those action potentials were important to learn about? When the action potential arrives at that axon terminal, it's going to help release those neurotransmitters, which then are going to communicate with the next cell. So here, before the neurotransmitter gets released, in the axon terminal, you have a sudden rise of calcium. And that sudden rise in calcium is going to stimulate those synaptic vesicles, the little vesicles I drew in, to merge with the presynaptic membrane. So that way they can release their neurotransmitters. So here we've got our terminal branch, which leads us to the axon terminal. Now here you can see we're going to have the action potential make its way down this axon terminal, and when it arrives at this axon terminal, it's going to open up voltage-gated calcium channels. Since the action potential is a voltage change, these are voltage-gated receptors. So they respond to that voltage change by opening up. And when they open up, calcium floods that axon terminal. So when calcium comes into the cell, this is going to cause these synaptic vesicles to then start to fuse towards this membrane. And when they fuse with the membrane, you can see they are made up of the same thing as the membrane is made up of. And if you remember, the membrane is made up of phospholipids. So they're made up of phospholipids as well, and they'll blend in with the membrane, and at the same time they release their neurotransmitter. So here we can see then when that neurotransmitter gets released, the neurotransmitter is going to go across the synaptic cleft, and it's going to bind to receptors on the postsynaptic neurons membrane. So when these receptors now have their neurotransmitter bound to them, this is going to allow them to open up. And when these receptors open up, these receptors then are going to allow ions to cross. Now here we see sodium crossing. Now when sodium crosses, we have seen the voltage on the inside is going to become more positive. So we're looking at one synapse right in there. And this is everything we see happen there. So now this cell is getting excited. This cell then is going to be able to generate an action potential. So now when this postsynaptic neuron generates an action potential, that action potential is going to be generated on its axon after the signal is passed in through the dendrite, as we see here, or the cell body, and the cell body integrates that information. Then the axon is going to generate an action potential, and this is then how cells will communicate with each other. Next, let's talk synaptic integration. A single neuron may receive many signals. It can receive excitatory signals, and if we talk excitatory signals, we'll see they're going to have a depolarizing effect. And what they're going to do is they're going to bring the neuron closer to threshold. Our inhibitory signals, they have a hyperpolarizing effect. They're going to hyperpolarize the membrane. And what that does is that takes us further away from threshold. And that's going to increase the potential's difference across the axon. So that takes us further away from threshold and less of a chance of having an action potential. And we can see synaptic integration is the summing up of excitatory and inhibitory signals. And this is going to determine whether an action potential will travel along an axon. So again, we can see various types of synapses. There are at least 25 neurotransmitters that have been identified. Acetylcholine and norepinephrine are the best known neurotransmitters. Both are excitatory neurotransmitters. Now here we can see once a neurotransmitter has been released into a synaptic cleft and initiated a response, then it gets removed from the cleft. In some synapses, it is inactivated by enzymes, where you can see, for example, acetylcholine, if that's the neurotransmitter released, acetylcholine is going to be broken down by acetylcholinesterase. It'll be broken down by acetylcholinesterase. Now, in other synapses, the presynaptic membrane is able to reabsorb the neurotransmitter as well. You have what we call reuptake there. So in other synapses, the presynaptic membrane reabsorbs, it reuptakes the neurotransmitter. And then here we can see the short existence of neurotransmitters prevents repeated stimulation or inhibition of the postsynaptic membrane. So it's got to be taken out. We want to get it out of there so we don't have continued stimulation or inhibition. Many drugs affect the nervous system by interfering with or potentiating neurotransmitters. Some drugs we'll see can enhance or block neurotransmitters release. Some can mimic the action of a neurotransmitter, and others can interfere with the removal from the cleft, where we can talk about messing with the enzyme. Next, this will take us to the next section where then we'll talk about the central nervous system. So when we talk central nervous system, we said the central nervous system is going to be made up of the brain and the spinal cord. And when we talk peripheral nervous system, you can see it's made up of nerves. You have cranial nerves and you have spinal nerves. The spinal cord and the brain, they make up the CNS, we said. And the CNS is going to receive sensory information. So sensory information is received by the CNS. And then motor control is going to be initiated here. The brain controls or influences bodily functions, some very important functions such as breathing, heart rate, body temperature, and blood pressure. And here we can see also the brain is going to be the source of emotions and higher mental functions. The brain and the spinal cord are protected by bone and then also by meninges. Meninges are protective membranes that are going to wrap around the brain and the spinal cord. And then we're going to have cerebral spinal fluid, CSF, that's going to fill the spaces between the meninges. And what it does is it cushions and it protects the central nervous system. The brain has interconnecting cavities, which are called the ventricles. And we'll see they're going to connect with the hollow central canal of the spinal cord, and they're going to be responsible for producing and then even storing some of that CSF. So you can see that CSF is going to make its way out of the ventricles and in between the meninges. The spinal cord is going to extend from the base of the brain, and it's going to make its way through a large opening of the skull. More specifically speaking, we saw the cranial bone, the occipital bone, the foramen magnum. It makes its way through the foramen magnum. Oh, we've got that listed there. Okay, and then it extends into the vertebral canal, which is going to be formed by the openings in the vertebra. Individual vertebrae are going to protect the spinal cord. These vertebrae are cushioned and separated by intervertebral discs, if you remember. Now, a cross-sectional anatomy of this spinal cord is going to reveal to us a central canal, which is going to contain CSF, not CFS, they put CFS, it's supposed to say CSF. Gray matter, which is going to be centrally located, and also it's going to take on the shape of a butterfly or the letter H. And then also we're going to have white matter, which is going to be surrounding that gray matter. And we'll look at that when we progress. So the gray matter is going to be shaped like the letter H or a butterfly. The gray matter is going to have a dorsal root and a ventral root to it. So when we talk the dorsal root, we'll see that's going to contain sensory fibers that are going to be entering gray matter. Ventral root, then we'll see, is going to have motor fibers that are going to be leaving the gray matter. And when we talk dorsal and ventral roots, they're going to join to form our spinal nerves. And then when we talk spinal nerves are going to be a part of our peripheral nervous system. Now the white matter we said is going to be found surrounding the gray matter. And this white matter is going to contain ascending and descending tracts. So it's going to contain ascending and descending tracts. Now when we talk ascending tracts, the ascending tracts are going to be located dorsally, and they're going to be sending information to the brain. And then our descending tracts, they're going to be located ventrally, and they send information from the brain to the spinal nerves. Many tracts cross over to the opposite side. And that's what leads to then the left side of the brain controlling right sides of the body and vice versa. So here we can see a nice vertebra, and in the vertebral canal we can appreciate the spinal cord. So here you've got the gray matter like the letter H, and then white matter surrounding it. And we've got meninges around that, and then you've got bone around it. Same thing we're going to have with the brain. And then here's your spinal nerve we can appreciate. So here's a spinal cord with all the meninges in greater detail. And we've got our dorsal root, which is again responsible for sending information towards the central nervous system, and the ventral root carrying it away towards the effectors. And then the dorsal root ganglia, you can see here, it's a collection of cell bodies. This is all the cell bodies of those sensory neurons. They're all collected right inside of there. Just like we saw in the beginning when we looked at these various neurons right back here. So if we get a whole bunch of sensory neurons, this is going to create then that ganglia. And that's what you're looking at. Right inside of here. And you can see the same thing right inside of here. Here's the spinal cord, the central canal. You can see right in the middle, central canal, right in the middle. That's where the CSF is going to be found, right inside the center. Now we talk functions of the spinal cord. The spinal cord is going to be involved with communication. It's going to be involved with communication between the brain and the peripheral nerves that are leaving the spinal cord. So we'll see when your hand is touched, sensory receptors are going to generate nerve impulses that are going to pass through sensory fibers to the spinal cord. The impulses then are going to travel up the ascending tracts to the brain. Then the brain is going to interpret this information. Okay, the sensory receptors in the hand were touched. Okay, this is a spider. Well, then the motor response obviously is going to be, wait, hold on, let me get my hand out of there. We were touching a nice, cute puppy dog. The response is going to be, okay, let's keep on touching the cute little puppy dog. So the impulse is going to travel up the ascending tracts of the brain, then the brain's going to interpret that information. And then we're going to have motor impulses from the brain pass down the descending tracts to the spinal cord. And they then go out to the muscles by way of motor fibers, saying, oh, get your hand out of there. Or, oh, that's so cute, keep touching it, in the example I gave. The spinal cord, we can see, is also going to behave as a center for thousands of reflexes. For example, nerves and muscles, they can respond very quickly to potentially dangerous stimuli. The stimulus is going to cause sensory receptors to send impulses then to the spinal cord. Interneurons are going to integrate this incoming data. Signals then are going to be relayed to our motor neurons. Then the reflex is completed when the skeletal muscles contract or the glands or the organs respond. So here we can see the spinal cord plays a similar role for the internal organs. Now if blood pressure were to fall, sensory receptors in the carotid arteries and the aorta are going to get stimulated. Then we're going to have impulses that are going to travel via sensory fibers to the spinal cord. They'll then travel up ascending tracts to the cardiovascular center of the brain. We'll see exactly where that's at. The brain then is going to send impulses down descending tracts to the spinal cord. And then we'll see motor impulses will travel via spinal nerves to the smooth muscle in the walls of the arteries, causing constriction to occur, helping to increase blood pressure. So when we talk about the brain, we'll see it's not just one structure. There's 4 major areas of the brain. First, we have the cerebrum, which will have at its center the 2 lateral ventricles. And then we have the diencephalon, which will have the 3rd ventricle. and then the cerebellum and the brain stem. And we'll see the brain stem will have the fourth ventricle found in relation to it. So here we can appreciate the cerebrum, all right inside of here. And here we've got one of the lateral ventricles right back in there. And here you've got the diencephalon. And then we'll have the third ventricle right in here. And here we can appreciate the cerebellum. And here we've got the brain stem. And here we've got our fourth ventricle. A nice picture of the two cerebral hemispheres we can see that make up the cerebrum. Now let's talk about the cerebrum. When we talk about the cerebrum is the largest portion of the human brain. We can see it's the last center to receive sensory input and carry out integration before voluntary motor responses. The cerebral hemispheres, the right and the left, are going to be divided from one another by the longitudinal fissure, but they're going to be connected by the corpus callosum. So here we can appreciate the two cerebral hemispheres of the cerebrum, and you can see they're divided by this longitudinal fissure we have here at the center. So here as we move forward then, let's talk more about the cerebrum. The cerebrum is going to be the largest portion of the human brain. It's the last center to receive sensory input and carry out integration before voluntary motor responses. Now the cerebral hemispheres, the right and the left, we said are divided by the longitudinal fissure. However, they are connected by the corpus callosum, which we can see right inside of here. This is all corpus callosum. This will connect the two hemispheres with one another. So if we were to kind of pry more into here, we would see then that connector is right down here. The cerebral hemispheres are going to be covered by a convoluted layer of gray matter, and we'll look at that in greater detail. And each hemisphere is divided into lobes by sulci. And here we can see the four lobes. Then we've got the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe of the brain. Now let's talk about that outer layer of gray matter, the cerebral cortex. The cerebral cortex is going to be a thin, highly convoluted outer layer of gray matter that's going to cover the cerebral hemispheres. Each fold we'll see is going to be called a gyrus. This region of the brain accounts for sensation, voluntary movement, and conscious thought. So first we can see frontal lobe, parietal lobe, occipital lobe, and the temporal lobe. All the different lobes. Now here we can see other structures. Other structures we can see then are going to be first the corpus callosum. Let's talk about that. The corpus callosum, we saw it helped to hold the two hemispheres together. It also helps them to communicate. And this corpus callosum is going to be a bridge of nerve tracts. It's a bridge of nerve tracts that allows the two hemispheres to communicate with one another. And here we can appreciate our sulci. So here we've got a whole bunch of sulci. These sulci are going to be shallow grooves. They're shallow grooves that are going to help divide the hemispheres into lobes. Also we have here gyri. Gyri, you can see, are these elevated ridges of tissue. All of these elevated ridges of tissue. Then we have the cerebral cortex. The cerebral cortex is going to be basically like the outer bark of the cerebral hemispheres. The cerebral cortex is a thin outer layer of gray matter that covers the cerebral hemispheres. The cerebral cortex contains motor areas, it contains sensory areas, and it contains association areas that we're able to see here in this picture. First, we've got the primary motor area. The primary motor area is found on the frontal lobe, just anterior to the central sulcus. So right in here we can appreciate the central sulcus that divides the frontal lobe from the parietal lobe. And we can see anterior to the central sulcus is our primary motor area. Now, when we talk primary motor area, we can see here the function of the primary motor area. Here is where voluntary commands to skeletal muscle are going to begin. Each body part is controlled by a specific section. And then we've got the primary somatosensory area. When we talk primary somatosensory area, it's found just posterior to the central sulcus, and sensory information from the skin and skeletal muscle arrives here. Here you can see also it's going to serve as the primary area for vision, hearing, taste, and smell. So here then we can see how the cortex of that primary motor area is going to control the skeletal muscle to these specific regions here. So when we talk this deep layer in here, the deep layer in here is going to start with the toes. And then we talk motor innervation to the ankles, it's down in there. And then as we move all the way up and around, you can see this will take us to the hand. And then coming around this way, it's the face, and then this way to the mouth and oral cavity area. So that's all motor innervation. Now when we talk sensory, here you can see we have a homunculus, is what this is called, a homunculus for sensory as well. Now we talk primary somatosensory area, we said the primary somatosensory area receives sensory information from the skin and skeletal muscles, and it's serving as the primary area for vision, hearing, taste, and smell. So here you can see these specific areas of the brain, or you can say these specific areas of the cortex of the brain, are going to provide sensory innervation to these specific areas of the body. So if we have a patient that suffers a stroke and they lose motor innervation to a specific area, we can kind of pinpoint that area of where it's going to be at. Next one, we have our association areas. Now, when we talk about the association areas here, they're going to be involved with integration. So here's where integration occurs and where memories are stored. The premotor area organizes functions for motor activities, such as walking and talking at the same time. The somatosensory association area, it processes and analyzes sensory information from the skin and muscles. Our visual association area, this is the area that associates new information with previously received information. The auditory association area performs the same functions with regards to sounds. Next then we can talk about our processing centers. They receive information from the other association areas and perform higher level analytical functions. Such as the prefrontal area, it uses information to reason and plan actions. This allows critical thinking and also allows us to formulate proper behaviors. Wernicke's area we have next. Wernicke's area is going to help us to understand written and spoken words. Broca's area, it directs motor actions of speech. Next then we can talk central white matter. It's composed mostly of cerebrum deep to the cortex. So it's found located deep to the cortex. And it's going to be composed of tracts of axons that take information between sensory, motor, and association areas. We also have our basal nuclei here. The basal nuclei are going to be masses of gray matter that are found deep within the cerebrum. And they integrate motor commands. Let's talk diencephalon. The diencephalon is going to be composed of the hypothalamus, the thalamus, and the epithalamus, which we'll see is going to contain the pineal gland. Now let's talk first hypothalamus. The hypothalamus is an integrating center that's going to help in homeostasis. It's going to regulate body temperature, our water balance, hunger, thirst, and sleep. The hypothalamus also controls our pituitary gland. It's the link between the nervous and the endocrine system. Next, when we talk thalamus, the thalamus is a sensory relay center. It receives all sensory input except for smell. And it integrates that information and sends it to the appropriate area in the cerebrum. It's also going to be involved in the arousal of the cerebrum, and it participates in higher mental functions, such as memory and emotional response. And then we can look at the pineal gland. The pineal gland is located in the epithalamus. It's located in the epithalamus and it secretes the hormone melatonin. Melatonin then regulates our normal sleep-wake cycle. Next, then we can move over to the cerebellum. The cerebellum, it receives sensory input from our joints, our muscles, and other sensory pathways about the present position of our body parts. It also receives motor output from the cerebral cortex and where these parts should be. The cerebellum integrates information and sends motor impulses to skeletal muscles by way of the brainstem. And it also functions to assure that we have smooth, coordinated, voluntary muscular movements. And it's involved with maintaining posture. When we talk brainstem, the brainstem is going to contain the midbrain, the pons, and the medulla oblongata, which we can also just refer to as the medulla. So let's talk first brainstem. The midbrain functions as the relay center for tracts passing between the cerebrum and cerebellum. It has reflex centers for visual, auditory, and tactile responses. The pons or the bridge we can also refer to it as, it contains bundles of axons traveling between the cerebellum and the rest of the CNS. And the pons is going to regulate our breathing rate along with the medulla oblongata. So we talk medulla oblongata, you can see it regulates vital functions such as heartbeat, breathing, and blood pressure. It also contains reflex centers for swallowing, coughing, vomiting, sneezing, and hiccuping. So if we ask where these reflex centers are found, you have to be able to mention the medulla oblongata. And here we can also appreciate the reticular activating system, RAS. It arouses the cerebrum, which causes alertness. The reticular activating system also contains reticular formation, which receives sensory signals that it sends up to higher centers and motor signals that it sends to the spinal cord. So let's move on to the peripheral nervous system. Now the peripheral nervous system we said is made up of nerves and ganglia. So we talk nerves. Nerves are going to be bundles of axons. So we get a whole bunch of axons, put them together, they make up a nerve. So when you say you're getting on my nerves, you can also say you're getting on my bundle of axons. Then we have our nerve fibers. Nerve fibers are going to have axons that occur in the nerves. And then we have our ganglia. Ganglia are swellings that are associated with the nerves, as we have seen. When we talk nerves, we have 12 pairs of cranial nerves, and these 12 pairs are going to be attached to the brain. Some of them are purely sensory nerves, some are purely motor, and some of them are going to be mixed nerves, meaning they have both sensory and motor functions. And they're going to be mainly concerned with the head, neck, and the face, these nerves are. The vagus nerve, we'll see one cranial nerve. It's going to extend to the thorax and the abdomen with branches to most internal organs. So that one's going to make its way a little bit further from the head, neck, and the face. Now we talk spinal nerves. Spinal nerves, we have 31 pairs. One way I remember that is Baskin-Robbins, 31 flavors of ice cream. We have 31 pairs of spinal nerves. So every time I think spinal nerves, I think Baskin-Robbins. So these 31 pairs emerge from the openings in the vertebral column between the vertebrae, those intervertebral foramina we saw. Each spinal nerve originates when two short branches or roots join together, the dorsal root and the ventral root we saw. All of these spinal nerves are mixed nerves. They're all mixed nerves. And we saw the cell bodies of our sensory neurons are going to be located in the dorsal root ganglia. And then when we talk ventral roots, our ventral roots are going to contain axons of motor neurons. So here we can appreciate our 12 pairs of cranial nerves. So here then we can appreciate cranial nerve number 1. Cranial nerve number 1, the olfactory nerve, is going to be responsible for taking information from our olfactory receptors then to the brain. And in cranial nerve number 2, we can see here the optic nerve. It takes information from the retina of the eye to the brain. It's involved with visual processing. And then number 3, we can see here Number 3, we've got our oculomotor nerve, and it's going to provide innervation to our eye muscles. Number 4, the trochlear nerve, same thing, provides innervation to our eye muscles. Number 5, then we can see here, number 5 is going to take information from the mouth and it sends information to our jaw muscles. Number 6 sends information to the eye muscles. So you can see CNS, eye muscles, right? Instead of the organ to CNS. And then we talk cranial nerve number 7. It sends information from our taste buds to the CNS, right? And then sends information from the CNS to our facial muscles and our glands. So that's what you have, to and from, to and from, to and from. And then number 8 we can see here, sends information from the inner ear to the CNS. And then number 9 we can see here, it sends information from the pharynx to the CNS and also from the CNS to the pharyngeal muscles. And then number 10 we can appreciate down here, 10 is going to send information from and to our internal organs. So it sends information from our internal organs to the CNS and from the CNS to our internal organs. And then number 11 we can see here, it sends information to the neck and back muscles from our CNS. And then number 12 is going to send information to our tongue muscles. So again, here we can appreciate our spinal cord, and we said the spinal nerves are made up of the dorsal and the ventral roots. So the dorsal and the ventral root come together to make up the spinal nerve. And that's our spinal nerves. And there's our cranial nerves. You can see the cranial nerves are a little bit more complex. Spinal nerves you've got there, we'll learn they just innervate in line as they come out. And they're mixed nerves. Here, spinal, we had some that were pure sensory, right? And some that were motor and some that were mixed. So we talk peripheral nervous system. When we talk peripheral nervous system, you'll see it gets divided up into the somatic system and the autonomic system. The somatic system is going to serve our skin, our skeletal muscles, and our tendons. The somatic system includes nerves that carry sensory information from the receptors to our central nervous system. It also carries motor commands away from the CNS to our skeletal muscles. Now, some actions are due to reflex actions, which are going to be automatic responses to a stimulus. A reflex occurs without having to think. And we'll see some actions are going to be voluntary. When we talk about the reflex arc is going to be a nerve pathway that carries out a reflex. Reflexes are programmed. They're built-in circuits that allow for protection and survival. They also enable the body to react swiftly to stimuli. They require no conscious control. For example, we can see here, if we were to get pricked by a pin, our sensory receptors that are found located in the skin would detect that, and they would transmit this information then via electrical potentials along the sensory neuron then to our interneurons in our central nervous system, where then the integration will occur. And once the integration occurs, then you can see they can excite the next cell, which is going to be the motor neuron. The motor neuron then can generate action potentials that are going to then excite our effector organs, which will then help us to move the pin away. So this is one example of a reflex arch. Next, then we have our autonomic system. Our autonomic system, it regulates the activity of cardiac and smooth muscle and our glands. Now, our autonomic nervous system has two divisions. We have first our sympathetic nervous system and we have our parasympathetic nervous system. Now, when we talk autonomic system, here then we can see we've got features in common to both divisions. They're both going to function automatically and usually involuntarily. They will both innervate all internal organs. They utilize two motor neurons that synapse at a ganglion for each signal, as we saw earlier. The first is the preganglionic neuron, and its cell body is in the CNS. The second is the postganglionic neuron, and its cell body is in the ganglion. Now, the sympathetic division is important during emergency situations. It mediates the fight or flight response. Now, when this fight or flight response gets mediated, what it does, it increases heart rate and contractility when you talk about the heart. And when you talk about the lungs, in the level of the lungs, it dilates the bronchi. And we look at it in relation to the digestive tract, we can see it inhibits the digestive tract simultaneously. This system activates the adrenal medulla to secrete hormones, epinephrine and norepinephrine. And then adrenaline and norepinephrine increase blood pressure, causing the heart to beat stronger and faster while constricting certain blood vessels. When we talk parasympathetic division, we'll see the parasympathetic division is going to include a few cranial nerves and fibers that arise from the sacral part of the spinal cord. This parasympathetic division is considered our housekeeper division, and it promotes all the internal responses referred to as rest and digest. So basically it promotes, you can see, food digestion, and it decreases our heart rate, to name a couple of tasks. The neurotransmitter that's going to be involved with the parasympathetic division here is going to be acetylcholine. So here's a nice picture that depicts to us both sympathetic and the parasympathetic divisions. So you can see our body is always going to be in one state or the other. You're either in a sympathetic state or in a parasympathetic state. Okay? In the sympathetic state, we said, it speeds up the heart, you can see. It inhibits tears. It dilates the pupils, right? We want to dilate the pupils so we can allow more light in case we have to fight or flight. Okay, so when you talk lungs, it dilates the air passages. We want to get more oxygen in, so that way in case we need to fight or flight. And now, parasympathetic division, you can see it does the opposite. It's going to stimulate tears. It constricts the pupils. It's going to slow the heart. It's going to constrict the bronchioles. So you can see, again, you're always in one state or the other. So you have to be able to understand what happens in relation to each of these organs if sympathetic activation occurs, and what happens in case parasympathetic activation occurs. So next here then we have a table. This table is going to compare somatic motor and autonomic motor pathways. So here we can see they're talking somatic motor pathway, and here we've got autonomic motor pathways.